So we'll get started. So thank you for joining this SADS Foundation Living with SADS webinar today, June 11th, titled Driven by You, Advancing What's Next in Long QT Syndrome Care. I'm Marcia Baker, the SADS Foundation Program Director. This webinar is being sponsored by Thrive Therapeutics. On behalf of the SADS Foundation, we thank Thrive Therapeutics for its grant support to make this webinar possible. And now I'd like to introduce you to our two speakers. Dr. Sam Sears is a clinical health psychologist and professor at East Carolina University, jointly appointed in the departments of psychology and cardiovascular sciences. He is internationally recognized for his work in the psychological care of patients with cardiac conditions, particularly those with implantable cardioverter defibrillators, or ICDs, and genetic heart diseases like long QT. And Sam is a longtime friend and colleague to the SADS Foundation. Joining Sam will be Dr. Amy Sehnert, the Chief Medical Officer of Thrive Therapeutics. Amy leads the company's clinical development efforts focused on novel treatments for cardiac conditions, including long QT syndrome. A pediatric cardiologist, Dr. Sehnert brings over 2 decades of experience in both patient care and clinical research, including previous development of an approved drug for rare genetic heart disease. This webinar will provide an overview of recent clinical study results from Thrive's research into a treatment for long QT and an update on a potential new treatment in development for long QT. We're honored that both Sam and Amy are joining us today. So we thank you, our community, for participating as well and hearing firsthand from Thrive representatives. Following the presentations, you'll have the opportunity to post your questions in the chat. Questions will be addressed at the end of the presentation. A reminder that this webinar is being recorded, so please keep your microphones muted during the presentation. The webinar recording link will be shared with all of you in a few days. Amy and Sam, I'll turn the microphone over to you now.
All right, wonderful. Thank you, Marcia, and hello, greetings to everyone from Montreal, Canada, where Thrive Therapeutics is based. I really welcome everybody who's joined and anyone who may have registered and listens to this recording at a later date. So tonight we're going to talk about long QT syndrome, and in particular, the 3 most prevalent genetic types, type 1, 2, and 3. I'm going to introduce you to serum and glucocorticoid regulated kinase, so-called SGK1, which is the therapeutic target that we at Thrive are relentlessly pursuing to see whether it might be a beneficial treatment in long QT syndrome. I'll talk a little bit about our Wave 1 clinical study results, which were recently presented at the Heart Rhythm Sessions in San Diego at the end of April. And what's next, what's coming, an effort called My QT Wave, and Sam will also be speaking to this. So patients are really why we're all here. They drive what we do. And this little girl, Isla Hutton, is truly the inspiration at Thrive Therapeutics. Unfortunately, Isla passed away from her disease just prior to her 7th birthday. But again, she inspires us every day, as well as her father, David Hutton, who has represented the patient community and supported the efforts. We know that long QT syndrome is more than the measurement of the QT interval on the EKG, and it's really about the whole person. Long QT syndrome is an inherited genetic disorder. Using Isla as the example, she had long QT syndrome type 3. There are no regulatory approved treatments for long QT syndrome. But in the clinic, we do use therapies, medical therapies, such as beta blockers, medical devices, such as implanted cardioverter defibrillators, surgeries, such as sympathetic nerve denervation, and sometimes another an antiarrhythmic drug called mexiletine. Isla was receiving all of those treatments at the time that she succumbed to her disease and experienced many of the symptoms. She had arrhythmic events, received shocks from her device, fainting, palpitations, in fact, also seizures. LQTS affects 1 in 2,500 individuals, and it really underscores the unmet medical need and drives our efforts to try to identify a therapy for this disease specifically. So what is the QT interval and why does it matter? Our hearts beat about 100,000 times a day, and every time they beat, they have to reset to be ready for the next beat. That interval of time when the heart is resetting for the next beat is measured as the QT interval on an EKG. When the QT interval is prolonged, it's not always ready for that next beat, but an early beat coming in can create an arrhythmic event known as torsade. So getting to the role of genetics in long QT syndrome, the genetics underpinning this disease were identified 30 years ago in 1995. And again, as I mentioned, LQT type 1, 2, and 3 are the first, second, and third most prevalent types of LQT. They are also sometimes referred to as channelopathies. And the reason for that is because the mutations occur in genes that encode channels for potassium and sodium ions to cross the cell membrane and create that heartbeat. So in LQT type 1, the mutated gene is called KCNQ1. It's a subunit of the potassium channel, and the effect is that it slows the outward current. Potassium ions need to go out of the cell to reset for the next heartbeat, and the mutation creates a slowing of that outward current. Similarly, in LQT type 2, it also affects a subunit of the potassium channel, and the mutated gene is called KCNH2. It also slows this outward current. In LQT type 3, it affects the sodium channel, and in that case, it's a so-called gain of function mutation that drives a persistent inward current of those sodium ions. And so therefore, again, it's prolonging the time the heart is taking to reset and be ready for that next beat. So why does QT interval matter? The risk of QTC prolongation beyond the normal range, it's real, and every 10 milliseconds of increase matters. This chart comes from a publication that is showing for every 10 millisecond QTC interval increase across the bottom from 461 milliseconds all the way to greater than 560 milliseconds, that there is a 15% increase in the risk to have a life-threatening and arrhythmic event in the next 5 years. So every 10 milliseconds matters. And another thing that this graph really shows is that for LQT type 2 and type 3, the risk of that is 130 to 157% higher than those with LQT type 1. So prolongation of the QT increases risk, and the type of LQT affects the risk. So what is the goal of treatment then? We know that beta blockers are used as a first-line therapy. They help to slow the heart rate, decrease the sympathetic tone, which can be helpful in this disease. The defibrillators are there to shock an arrhythmia that is persisting and is a sudden arrhythmic event. And what about shortening the QTC? I did mention the drug mexiletine is used, which is a so-called sodium channel blocker. It blocks the channel, the sodium channel. But we've been pursuing SGK1 inhibition to have the effect of reducing the QTC, which in turn would reduce the risk for a sudden arrhythmic event. So why SGK1 inhibition? This is just kind of a blown up diagram to help show what's going on across the cell membrane. Looking on the right, you're inside the cell, and on the left, you're outside the cell. So those potassium ions at the top are trying to get out of the cell in order to reset. And on the bottom, the sodium ions are going in the cell. SGK1 is a kinase. What's a kinase? It's an enzyme that activates other proteins. And in this case, it's keeping the door open on the sodium channel. So the sodium ions just keep coming across, and they need to stop coming across in order, again, to be ready for that next heartbeat. So inhibiting SGK1 with an SGK1 inhibitor, our first treatment that was developed was called LQT-1213. What is that? That's a drug. That's a drug that you would take by mouth, and then we can measure what effect does that have on your QTC. Now, I know the SADS Foundation in recent months has had a webinar about clinical research. And so based on this foundation of science, of understanding this ion trafficking in the cell and how we might inhibit it to shorten the QT interval, phase 1 studies were done. Healthy volunteers volunteered to help us get to a point where we could get to the clinic and treat patients with LQT type 2 and 3 with this drug to see if it would have the effect that we were seeking to shorten the QT interval. So this presentation about that study, as I just mentioned, was presented in San Diego at the Heart Rhythm meetings. And we shared it with the attendees, the physicians and academic researchers there. The purpose of the study was to evaluate the effect of SGK1 inhibition with this drug that had been developed, LQT-1213, on the QTC interval in patients with genetically confirmed type 2 or type 3 patients. If any of these patients are on this webinar or ever listen, thank you. Can't thank you enough for having participated in this study and undergone 2 1/3 days of dosing. Not only that, came to an inpatient setting over a number of days in order to participate. It's really what drives the progress forward. So what was the study? We called it Wave 1 Part 2. There were 6 subjects in each of 2 dose groups. So we tried a low dose and a little bit higher dose of the drug. The patients were adults over 18 and had QTCF measures at baseline that were over 480 milliseconds. We'll see that I'll give you the baseline characteristics of the patients in a moment. So what the patients did was they were admitted to this unit. And the first day, they had a Holter monitor applied, and they took a placebo, which is, it was Gatorade. That was the placebo. And then for 3 days, they took the drug 3 times a day, 8 hours apart. And they had a Holter monitor applied on the first day of dosing and the last day of dosing. They took a total of 7 doses, 3 on day 1, 3 on day 2, and 1 on the last day of dosing. And then we took that information from the Holter monitors and we evaluated what effect did it have on the QTC, Importantly, we were also looking to see whether it was a safe experience. So who were these patients? There were 9 females. The average age was 41. There were 9 patients with LQT type 2 and 3 with LQT type 3. The baseline average QTC was 521 milliseconds, so pretty significant prolongation of the QT. And 11 were on beta blockers, and 11 had ICDs. So this treatment was received on top of that standard of care. And so just at a high level to conclude on what the findings were, was that in these patients, this oral treatment was well tolerated. We did not see any moderate or severe or significant adverse effects. In the high dose cohort, we saw meaningful reductions in the QTC interval. And I've put the numbers there associated with it, but really the key takeaway is that in this early study, after a few days of dosing in these patients, we did see a reduction in the QTC interval in a way that provides encouragement to proceed and to continue the results of SGK1 inhibition. And we'll be initially doing that in LQT2 and LQT3. We really look forward to having the next phase of this study come to you. Hope to be back in the future to tell you about that. We will have a so-called Wave 2 study to proceed testing this drug in LQT patients. And in the meantime, we're planning to launch this this observational effort and pivotal patient experience called MyQT Wave. Again, we see long QT patients as whole people and whole persons, and Sam's going to tell you more about how we're going to, within this study, try to better understand the lived experience with long QT syndrome. It's very important to understand when you're trying to affect it with a new treatment and really make a change in people's lives. So with that, I'm going to turn it over to Dr. Sears. Sam?
Hey, well, good evening, everybody. Great to be back on the SADS channel, if you will. And I'm excited to talk about this study. Clearly, living with long QT syndrome has significant challenges. And it goes beyond simply creating a magic medicine. It goes beyond that. As important as that is, it goes into the idea that real people seek comprehensive solutions. And when we think about the patient priorities and we think about long QT syndrome, we've created this opportunity together with Thrive to, I think, further our understanding and about living with this condition. And we'll start with the idea of MyQT Wave. That's the title of our study. Next slide, please. Now, when you think about surfers, surfing, right? And I grew up in Florida, and I know a little bit about surfing, not a lot, but when you watch surfers, there's some grace there, isn't there? There's some grace about, look at the power of the wave, and yet finding a way to surf it, finding a way to go with the flow and somehow stand tall. Honestly, as we take care of patients like you in SADS conditions, I see surfing. I see people and admire that ability to stand tall even when there are challenges, stand tall when there are forces around you. And I think understanding that begins this process of MyQT Wave. Next slide. So I'd like to start with talking a little bit about what we know as a psychologist in this area for over 30 years now. What do we know about the clinical experience, the patient experience of living with long QT, and then talk about the study about what's next. Next slide, please. So we start out with the idea that all patients with long QT and their families have some degree of risk for psychological distress. It's frankly not normal to worry about the QT interval of your heart. That by itself sets patients and families aside. And the ambiguity that we've seen over the years about what's going to happen. Am I going to be okay? Am I going to live a full lifespan? These ambiguities are part and parcel to the experience of this disease. And families, of course, experience that. It's not just the individual, but the cascade relationships of this
Can't hear anything.
genetic condition. So collectively, this idea of medical stress and living with psychological stress is part of living with long QT. Next slide, please. So in one of the larger studies that we have available to us, the group at Hopkins looked at both ARVC patients and long QT patients and showed that approximately a third of patients with these channelopathies or ARVC had anxiety that was problematic. So one in three. And approximately 16% had this experience of depression, both of which are elevated compared to norms. Certainly the depression, less elevated, the rate of anxiety is quite elevated for compared to same age norms. Next slide, please. And we know that the experience of cardiac arrest, the experience of a loved one experiencing a cardiac arrest is strongly associated with a traumatic stress response, a PTSD response. And you can see some of the data here in this Australian study with 103 families that faced a cardiac arrest, some aborted, some resulting in death. But in both sets of circumstances, patients experiencing the high rates, that is, the families experiencing high rates of PTSD. Next slide. And as I mentioned, grief is also part of that picture, prolonged grief about the loss. I think everybody can see that first slide and recognize how powerful the losses that this condition provides. We admire and honor that your ability to get through these things, to surf your way through these despite the power and threats and risks that go with this. And prolonged grief is part of that picture at times. Next slide. So we also got, I think, the best set of data from a patient-focused group led by SADS last year in September. So this is very recent data, and it involved a group of patients that came together and answered specific questions to allow for some survey responses. Next slide. The highlight of the report involved things that I think everybody on this call probably recognizes, that long QT syndrome is associated with a fear and worry, particularly about the future. That the most burdensome health problems that go with long QT syndrome were actually anxiety and depression, and that resulted in a diminished quality of life overall. And while the treatments have been able to save some lives, they clearly aren't curative and they aren't the whole story. And so the idea of developing future treatments was desirable. And we'll look at some of the challenges that patients reported here in the next slide. So in the most troublesome, when patients were asked what was the most troublesome concerns they had, as I noted, anxiety and depression, with over half of the patients saying that anxiety and depression was the most troublesome concern. But you also see the specific cardiac concerns of lightheadedness or fainting, cardiac arrest, or potentially fatigue and sleepiness, which would be associated with some of the current medications that we would use to manage long QT syndrome. And you can see some of the other ones on down to fear of shocks, with 26% of those, obviously with ICDs, reporting fear of shocks. Next slide. And the impact on daily life is one of the ways that patients express themselves, saying that this condition primarily targeted their stamina, that these young people, in this case, a relatively young sample, reporting that not much stamina, not much ability to go further and sort of keep up with same age peers in terms of energy and activity levels. And you can see that many had to choose not to engage in sports, but things like biking or playing other sports. You can see just on down that there are a number of ways in which this condition impinges on their daily life and their choices of what they could do each day. Next slide, please. And the medications or therapies that are available, what changes were asked of them? Well, you can see the number one change asked of them was medication use, beta blockers used to control heart rate or slow heart rate, ICD implantation, 59%, and then on down, again, avoiding sports. Look at third, seeking psychotherapy or counseling, almost a third experiencing and needing antidepressants or anti-anxiety, and on down. But you can see that there were a number of changes that were necessary because of this condition. Next slide. So an ideal treatment, when asked about the things that an ideal treatment would provide, preventing arrhythmias was number one, preventing that known threat from long QT syndrome. Secondly, a treatment that would increase energy or not at least take away energy, a treatment that would improve daily functioning, and finally allow participation in sports and allow for better handling of emotional symptoms that go along with this condition. Next slide. So we're not at the beginning of this. Long QT syndrome, recognized as an arrhythmic syndrome, a channelopathy, recognizing that this is a challenging condition psychologically, recognizing this is a condition that affects more than just the index patient, recognizing this is a condition that has a number of lifestyle implications because of the treatments necessary to protect our family members. So what do we do next, and what do we need to know now? Next slide, please. Well, working together with Thrive, about 3 years ago, Thrive came to me and said, we'd like to think about how to help this whole group of patients that have long QT syndrome. And we want to know, how could we design a treatment and understand a treatment that would truly make a difference, that would change the way that patients experience and live with this condition. And together with other colleagues, we suggested some things and some areas to study. And over the last 3 to 5 months or so, we've been talking about this study going forward now. It's a non-interventional study, an observational effort funded by Thrive, driven by cardiologists and psychologists to understand exactly the experience of living with long QT type 2 and 3. Next slide, please. The study highlight is one I've never been a part of before. Despite having doing hundreds of studies, this study would be a 6-month remote study with no clinic visits. Instead, we would rely on an Apple phone to query and ask questions about how you're doing at various times, short surveys via that. We would also have daily symptom tracking, so letting us know if you experience any symptoms like lightheadedness or fatigue. And further, we would have regular ECGs provided from an Apple Watch. We think this is an interesting way of using this new technology. It's certainly more convenient. Next slide. So who's eligible, right? So we're looking for adults 18 years and older and have a confirmed long QT2 or long QT3 diagnosis from genetic testing or a doctor's letter confirming that the QTC corrected interval is greater than 480 milliseconds. We hope to roll this study further into long QT1 patients at a later date, but we'll start with these two particular groups of long QT2 and 3. Next slide. So the goal of this study is to describe the physical and emotional symptoms of these two subtypes, to describe its impact on daily activities, with everything spanning sleep and physical activity to stress and anxiety, and to assess those things, and then to use the Apple Watch to give us some information, objective data about physical activity levels and even some cardiac-related information. Next slide. The focus that we recommended was to go beyond simply a measure of just how are you doing, and to try to use standardized metrics of health status and disease burden, to look at standardized ways in which perceptions of your illness and the daily activities of managing your health are engaged, and then finally the psychological and psychosocial components, how anxiety, stress, depression, how these things interact in such a way to affect your life in various ways and to target quality of life in particular. Because ultimately what we want to do is create therapies that do impact quality of life broadly, that would encompass all of these things. But we want to be sensitive to exactly the components so that we have an excellent description of exactly what we're targeting and how to target it. Next slide, please. So using these tools of both a smartphone and a smartwatch, together with a sampling methodology that would come about at regular intervals over the course of 6 months, so that we would have much more day-to-day functioning, not just how you felt in the clinic one day, not just how you reported it at every now and then, but to truly get a descriptive experience of living with these conditions. This is what we target. This opportunity is unique. This will provide us with the gold standard of data about taking care and understanding the condition of long QT2 and 3. Next slide, please. And so with that, I think this opportunity to engage around this project will be a great one. The timeline remains somewhat sketched out, but we expect this study would be able to begin at the beginning of September and would flow through a 6-month period. So that'll be something we all look forward to as we get final regulatory approvals and complete the methodology of this study in order to roll it out. And with that, I'll conclude and open for questions.
Thank you very much, Sam and Amy. We appreciate your presentations. And I would invite anyone who posts their questions in our chat feature to everyone, and we'll be able to see those.
Well, while we wait for a question, oh, here's one. All right.
Okay, Amy, were any of the patients children or were they all adults?
All adults, and the average age was 41. So this was 18 and above. We are looking to include potentially patients 15 and above for the next study. And we do hope through the course of the development that we could eventually conduct a pediatric specific trial.
Okay, thank you. At some point, will the study be available to those with long QT2 and 3 who have a QTC of less than 480?
That's a great question. I think that we really will need the data from the population that are greater than 480 to really understand that magnitude of effect and determine whether that would make any sense to go below that level to further decrease. But really, that's the point where more of the risk begins to increase above 480.
Okay, thank you.
So I want to clarify her answer there because I think that's an important one. Because I think the spirit of the question was trying to, hey, I want to be in this trial. But I think, Amy, one of the things that you've taught me is that the effect of the medication below that point is still there, but in order to be able to prove the efficacy of the medicine, we will be able to flatten or reduce those to get to those important clinical markers of effectiveness with somebody where the long QT is wide or longer, right?
Yeah, that's correct. And even in the current study, those who were longer, over 500, actually had more of a decrease in effect. And so certainly, again, with every 10 millisecond increase, that's where the risk is increasing. So we want to balance, the treatment benefit with the need, which is greatest
exactly right.
at
So,
the higher end. Mm-hmm.
And the idea would be too that if the QTC is lower, the risk is lower, or at least potentially lower, and so you are just going for high risk first because this is a medicine that we've got to go through regulation on, right? So we've got to prove it's both its safety and its effectiveness or efficacy. So yeah, we're excited about what this might give to our entire population, but we got to start with this particular subpopulation. Is that right?
Yeah.
Okay, Sam, would you consider pediatrics as part of your study?
You know, this was something we had a long discussion about. As of right now, because of its remoteness, we're staying with adults because there's different ethical considerations with people less than 18 that involve some legal and regulatory aspects. So I think right now we're getting started with just the adults. We definitely want to get toward the younger people, but we can't do that quite yet.
Okay, thank you. Is the study only for US patients?
Yes.
Are we hoping for
Yeah.
any Canadians to be involved or not, or other countries?
Yeah, this will be IRB approved and US focused.
Okay, thank
Mhm.
you. When would it potentially roll out to long QT type 1 patients?
The MyQT wave, I presume, is yeah.
I think so.
Well, I can speak to on the I can't give you an exact time frame when that would be open to the LQT1. We have some enrollment goals for the LQT2 and 3. We absolutely will keep you posted. And if you do have LQT1, we still want to know your story, who you are, and as we evolve, how we can begin to collect information from patients from LQT1. What we don't know is if the results across the subtypes will be different in any way. We do know there's some differences in risk, some differences in response to treatment, but stay tuned and keep in touch with us, and we absolutely will make sure to let everyone know when we're able to open it up to LQT1.
Okay, thank you. Would someone be able to come off beta blockers with this medication?
It's another great question that's going to require a lot of longer term research to really see. But keep in mind that really what the drugs are doing are two different things. So the beta blocker is reducing that heart rate, that sympathetic tone, focusing on that rate. The SGK1 inhibition is going to be focusing on the rhythm. So whether or not, that we are starting on top of that standard of care, and it would take quite a bit of future study to determine whether withdrawing the beta blocker, that the results would be the same with only a monotherapy.
Okay.
Yeah, I really like that answer there because the beta blocker is really at the rate and this is getting at the rhythm. I like that. I think that's really clear because we're still going to need to, or potentially still going to need to slow the rate for other reasons. And so I like that answer a lot because that makes it clear when you think about the heartbeat as both rate and rhythm.
Mhm.
And of course, the long QT speaking to the rhythm. And so the medicine then targeting that as well.
Okay, we have a clarifying question about the less than 480. She was referring to the MyQT wave study, not the medication
Gotcha.
study,
Yeah.
and sharing the side effects of treatment despite being well beta blocked below, and it's 480. Not sure.
Got you. Understood. So yeah,
Okay.
because we mentioned that the eligibility right now for MyQT wave is over 480. So with again, thank you for that feedback. We taking it back because we do see this as kind of an evolving process over time, and so it's another category that right now we're not including initially, but could consider because to your point, you're still on the beta blocker. And that's what's got you in that range. So thanks for that question. We'll add it to our list.
Okay. If this medication passes the clinical trials, how long would it be before it is commercially available?
Mhm. Well, even after a pivotal study, when a drug application is filed to the regulatory agency, it takes another year beyond that. So we're at least, 2, 3, 4 years away from a drug that would be approved and be available in the clinic.
Okay, thank you. So if a patient has an ICD, might they be able to have it removed at the time of required replacement with this treatment?
Mhm. It's a little bit like the beta blocker answer in terms of, okay, the beta blocker is doing heart rate. The ICD is there's a protection mechanism to shock out of that arrhythmia. The SGK1 inhibition is going to be reducing QTC, which we believe will reduce the risk for arrhythmia. But again, it would take a lot more research and evidence to be convincing that it was so much so that you now don't need your defibrillator. But I think that the drug is not going to shock you out of that rhythm. So right now, the device is really the only kind of life-saving event in the moment of that arrhythmia, which can be unpredictable, despite the medical treatments we have right now. And certainly that would be a goal for the future. And it's a great question. But for sure, we can't say anything about that at this point in terms of making that type of medical decision.
Okay, thank you. This person has a son that's treated at Duke University. Is Duke going to be involved in any of these trials in any way, or in North Carolina or East Carolina as part of the next trial phase?
So the MyQT wave is going to be, that's why we chose Driven by You. It's really a patient self-referred to the trial for enrollment. Of course, you will need to provide the confirmation of the genotype or doctor's letter, as Sam mentioned, looking at the eligibility that we've talked about. But we do very much hope to be working with Duke and or other sites in North Carolina as we get to the next Wave 2 interventional study.
Okay, thank you.
And I would just encourage everyone to indicate, either via SADS or as we provide the contact information for this study, what's your story? What are you interested in, and how can we best keep you informed about our work as we continue down this path?
Okay, I have a question. Why is type 1 not included in the upcoming study looking at mental health?
Sam, do you want to?
Yeah, I think we had to start with where the medication is going to start. I think first is that we're targeting long QT 2 and 3 with the medication first, and so my QT wave also follows suit with that. And I think as we get a little bit more experience with the trial, long QT1 would be a much larger group as well. So we are able to start with the smaller 2 groups and then move to that in January, or we think early next year, next calendar year. That's where we target that. We obviously have to get started with the first 2 groups, but we think that's important too. We know that this is, from a patient experience standpoint, we expect them to be more similar than different, but there are some differences that would be true and would be relevant here. So we do understand, well, in fact, we love that people want to get into the studies, because this is how we're going to understand things. We are where we are today. The work that many people have spent decades on to get us to a point where poor Amy's got to describe how an SGK1 works, talking about all these cells and things. Well, thank goodness we have the scientific community working on this. We want to go faster. we all want to go faster. We want to get this thing. If this works, let's get it to you as soon as possible. Same thing with the studies. You know, I want to study everyone, but we got to get started somewhere and then roll into the next. So I love the enthusiasm because I think that's what it drives us. I mean, these are people have committed their life to understanding this condition, and all of us, we experience it in clinic every day, and so this is important to all of us. So we'll get there. We just can't get there yet.
Okay, thank you, Sam. Here's another question. How does this medication, this treatment medication, compare to mexiletine?
Good question.
Yeah, no, that's a great question. So, if you followed the cartoons with the ions moving in and out of the cell, the sodium ions are moving into the cell, and that's during the time where the heart is resetting the switch to get ready for the next heartbeat. And mexiletine works by plugging that hole, plugging the sodium channel from the outside of the cell so that sodium ions can't come back in. It is an antiarrhythmic, and the contrast just out of the gate is that SGK1 is inside the cell. We're not plugging that channel. We're inhibiting an activity that's keeping it open. So mechanistically, they're functioning in two different ways. The mexiletine is dosed 3 times a day. We're targeting to get to a twice a day dosing. Also, of course, mexiletine has, as many drugs do, side effects, pretty well known side effects that we need to evaluate with SGK1, and then we'll be able to understand kind of the safety and side effect profile better. The other thing is that mexiletine is an antiarrhythmic, and as hard as everyone has worked on the arrhythmia area, many of the antiarrhythmics available today actually have a side effect of being pro-arrhythmic. We also hope to be able to avoid that type of side effect. But from the cellular level, you're plugging the hole on the outside, or you're closing the leak from the inside of the cell. They are mechanistically doing something different there to try to achieve a similar goal around shortening that QTC interval.
Okay, thank you, Amy. Thank you very much. And I think that concludes our questions. So I would like to thank both Amy and Sam for their presentations today, and thank all of you to participating today. And so have a good evening. Thank you very much.
Thank you so much.
Thank you.